The SageELF, whole sample fractionation for DNA sequencing and proteomics studies. Get the most comprehensive view of your DNA or protein. The SageELF is the first of its kind technology that can automatically select and collect multiple fractions from a single DNA or protein sample, making it ideal for precious samples. Using automated preparative electrophoresis, one DNA sample or one protein sample can be parsed into 12 contiguous size fractions. ELF is short for Electrophoretic Lateral Fractionator. The system features a disposable precast agarose gel cassette onto which a sheared DNA or complex protein sample is loaded. 1 or 2 samples may be fractionated during a run. The ELF cassette features a separation column in which DNA or proteins are separated by size using parameters set by the user. When the separation is complete, a second set of laterally positioned electrodes is activated, and fractions are electroeluted into 12 physically separate membrane-bound wells. The fractions may then be removed by pipette. To set or change fractionation range, users choose a value for a well in the software representing the median size for the fraction to be collected in that well. The instrument will estimate the expected median sizes for the remaining fractions. For some applications, a fluorescently labeled internal marker can be used to improve the accuracy and reproducibility of fractionation. The SageELF allows researchers to select multiple insert-sized libraries from a single DNA sample. This provides an unprecedented tool to study genomic rearrangements using mate pair sequencing. For RNA studies, the identification of new splice variants and isoforms can be enabled by segregating size ranges with high expression levels from those with lower expression levels. And for scientists and clinicians who work with precious samples, the SageELF provides the opportunity to construct libraries for sequencing while preserving the remaining fractions for later study. Equipped with pulsed field electrophoresis, the system is capable of resolving DNA at 100 kilobases and above for third-generation single-molecule sequencing approaches. For protein studies, the SageELF reduces the complexity of protein samples in a labor-saving and reproducible manner. Eluted proteins can then be analyzed by mass spectrometry or pooled for affinity capture or enrichment of rare proteins. The SageELF. Use every part of your sample and get the most comprehensive view possible.